Hello, cell biology fans. Sitting in my office the afternoon before the holiday, kicking things, knocking things over. And I thought I would do this quick lecture for you on the cell cycle. A lot of this should be something that you remember from 107 or other classes. Hopefully none of this is too new. Some of it will be new, the part on the microtubules, so pay attention, okay? The idea of the cell cycle is that a significant portion of the cell cycle is when the cell is just out there doing its normal thing, and at some point it needs to replicate the DNA, and it does that in S phase. Now, what I like and what I don't like about this particular chart on this page is you see that 23 hours is interphase. Interphase is the time between rounds of division. So you have interphase, then you have mitosis, then you have interphase, then you have mitosis. Interphase consists of the G1 phase, S phase, and G2 phase. And S phase is really the synthesis phase. And if you think about that, it's synthesizing the DNA. So you're doing replication of DNA, and you're also going to synthesize another important feature of mitosis, which is, anybody? The MTOC. And the MTOC is really the Centrioles, or the Centrosome. Okay, so this is what we're going to talk about as far as the cell cycle goes. Replication really means that we're making copies of DNA, and you should all know that replication of DNA is semi-conservative, which means that you always have an old strand paired with a new strand. The process of mitosis is how we actually segregate those sister chromatids from one another during the process of pulling the chromosomes apart. So mitosis is separating the chromosomes. Cytokinesis is actually the separation of the two daughter cells. And sometimes these aren't always linked together. In some cases, you actually see that you get replication of DNA, separation of chromosomes, but the two daughter cells don't actually get pulled apart. And there are several examples of that. The early stages of endosperm development in plants, syncytia in Drosophila. Syncytia are cells that have multiple nuclei, so we often see this when cells are virally infected. And there are some polyploid tissues, which we've already talked about. Skeletal muscle cells have multiple nuclei in an individual cell. So this is not uncommon. It's something that actually happens on a relatively frequent basis. If this happens in any other kind of cell, it is detrimental to the growth of the organism, and those cells will automatically undergo the process of apoptosis. So keep in mind cytokinesis. We'll talk about that at the very end. That's the separation of two daughter cells. The major players in this, who is actually doing all of the work, so you have chromosomes that need to be replicated. You have the centrioles, which are part of the spindle complex. The actual process of cytokinesis involves a contractile ring, which is made of actin filaments, so actin and myosin. That's in animal cells. In plant cells, the separation of the two cells from one another is due to a complex called the phragmoplast. And what's interesting about that is it's actually GOLGI is going to start spitting out membrane, and the membrane-bound vesicles that are spit out from the GOLGI create a plant cell wall. I'm sorry, not a cell wall, but a cell membrane between the two daughter cells. All right, so we're going to talk about each of these in turn. I already showed you this picture of what DNA from a nucleus really looks like during normal non-mitosis phase. And so during interphase, the DNA is this long spaghetti-like strands that you see on the left in panel A that is emanating from the nucleus. On the right is what you see at the time of mitosis, during the phases of mitosis: prophase, metaphase, anaphase, telophase. Okay, so that's what it looks like when we actually get down to the nuts and bolts of doing the separation of DNA during mitosis. You should already know that the way we get these levels of compaction, you start out with regular double-stranded DNA. They're anti-parallel. They wrap around nucleosomes. Those are made up of H2A, H2B, H3, and H4. That's the beads on a string structure, right? Those tend to wrap around themselves, making the 30 nanometer fiber, and after the 30 nanometer fiber we start seeing these loops form in the DNA, and I'm going to tell you how those loops begin to form. There's a protein that helps do that. Ultimately, all of this gets jam-packed into the nucleus, and all of this has to be made very tightly during the process of mitosis so that we don't have big long strands of spaghetti that get wrapped around one another and broken during the process of pulling sister chromatids apart. So the proteins, a significant number of the proteins that contribute to this are called condensins and cohesins. Yet, once again, another brilliant name for proteins. They have a very similar structure. So in panel A, on the left of this image, you see a protein complex. It's made up of I hope you can see that it has an N-terminal head and a C-terminal head. There are two of these. They're anti-parallel to one another. So there's two globular heads that are linked to one another through this coiled coil domain, and at the very point of this molecule you have another complex shown in blue called the Hinge region. And what's interesting about this is these globular heads are what bind to DNA, and they're associated with ATP. So what do you think ATP is going to do? Binding and hydrolysis of ATP changes the conformation, which changes the way it links with the DNA, and this is what allows for them to create structure. So condensins and cohesins have this structure. So I think of them as little V's and think of them as being able to grab DNA. So, right, if my two fingers can grab DNA at different parts, dink, right? In panel B, what they're showing you is that it is the cohesins that link one daughter strand to the other daughter strand. That's what keeps the daughter strands really close to one another. Whereas it's the condensins, which in panel C create the loops that we see in the third level of structure of DNA. So condensins and cohesins play a significant role in the packaging of DNA during mitosis. In this fluorescent micrograph, you can see in both panel A and panel B pink, and the pink is staining for, I believe it is condensins. So the condensins are what are creating the loop structure, and you can see them the whole length of the sister chromatids. All right, you still see pink even when the cohesins, so in panel B, the links between the sister chromatids have been released. All right, now you can see the sister chromatids, but you can still see that there's a significant amount of condensins in this structure. So there's a lot of proteins associated with getting condensed chromosomes so that we don't have anything messed up during mitosis. All right, so the centrioles. The centrioles are absolutely required to generate the spindle complex. The centrioles are part of the centrosome. The centrosome has two centrioles. Now, during the S phase, the centrioles are duplicated, and you end up having two centrosomes. Those centrosomes will migrate to both ends of the cell, and they generate microtubules that link out to the middle and grab onto the chromosomes at the metaphase plate, and they also go the other direction and bind to the plasma membrane on the outside of the cells. This particular image is showing you once you have the pulling of the chromosomes, the sister chromatids apart, another complex forms around the metaphase plate, and it's called the contractile ring. The contractile ring is made up of actin and myosins, and it basically is a ring that squeezes around the two cells and ends up pinching the two cells off from one another. So these are myosins that bind to two different actins and pull them close together and create this ability to pinch the two cells apart. So that's what happens during cytokinesis. The centrosome, right, it contains two centrioles, and in this diagram, so on the top what they're showing you is an EM picture of two centrosomes. And the bottom is actually trying to mimic exactly what's in the top picture in cartoon form. So what they're showing you is that there's a pair of centrioles on the right and that they are at perpendicular angles to one another. Some of you might ask why. It's a great question. I'm not sure the answer is known, but they always are in this particular conformation. So there's also a pair of Centrioles on the left-hand side, and you can see that there are microtubules. Some of them are very small in the middle. Those are probably just being polymerized or nucleated, and then there are some longer ones that emanate out from this structure. You actually have to have two of these in a cell to make a full spindle complex. So just remember, the formation of the second centrosome occurs during the S phase, the same time that you're duplicating the DNA as well. Okay? Alright, so let's talk about the particular phases and what you need to know as far as the phases go. The easiest way to remember all of the phases in order is IPPMAT. It just works for me. I is interphase, PPMAT. Prophase, prometaphase, metaphase, anaphase, telophase. I'll tell you how I remember it. The only way I remember it is PPMAT. MAT, the M I know is metaphase. Meta means middle. Okay, so then you have two P's, and which P is going to be before metaphase? It's going to be the prometaphase. So then the only other choice for P is prophase. That's how I remember it. Anaphase and telophase just come naturally for me. I stands for interphase. That's G1, S, and G2. Remember, G1, S, and G2 take up most of the time during a day. So let's say if a cell duplicates itself in 24 hours, 23 hours of that's going to be spent in interphase. 1 hour is going to be spent doing PPMAT, right? Prophase, prometaphase, metaphase, anaphase, telophase. And cytokinesis is the final step where we do the pinching off and separate the two daughter cells. So if you look at this diagram, it really is very simple, right? You see during S and G2, you have the formation, right? In the nucleus, you can't really see anything because the chromosomes haven't condensed yet to their fully condensed structure where we need to pull them apart. You can't really even see the nuclei. You can see that there are two centrosomes, but they haven't actually pulled apart from one another. And as you go through early prophase and prophase, you begin to see the condensation of chromosomes. The chromosomes actually become visible. You can see the structures, and during this transition from early prophase to prophase, obviously to metaphase, you have to have those centrosomes actually get to the very ends of the cell. All right, and obviously at metaphase, all your chromosomes are lined up on the metaphase plate. So right then they're at the poles. The centrosomes are at the poles of the cells, and they're called asters. What's an aster? An aster is like a star. And so they were given this name because they look like asters, because they have all these things emanating out from them like a star might. Okay, so you'll see that word asters come up again. So these immunofluorescence images basically show you exactly what we saw in the cartoon. During interphase, in the blue nucleus, you really can't see nuclei. The nuclei are not very visible, and only when you start getting into prophase, right? So you look at early prophase, you look at late prophase. Okay, maybe you can see some nuclei, maybe you can't. Once you get to prometaphase, all right, you can definitely see chromosomes. I hope that in number 3, you can see the centrosomes. So the centrosomes are in the blue, and they're actually probably behind the blue or above the blue, right? They're not in the nucleus. They're actually moving to the poles, so they're either above it or behind it. And you can see all the microtubules emanating out from there. in panel 4, you can really see the centrosomes separated, and you can see the yellow. The yellow is staining for the centrosomes, and the green is staining for the microtubules. Okay, and as you continue through this, at metaphase, you have the chromosomes lined up on the metaphase plate, and they're being attached by the Kinetochore Microtubules, and Anaphase is the signal to start pulling. And this is very, very dynamic. All of a sudden, they'll line up, they'll jiggle around, there's some pushing and pulling of the chromosomes this way, this way, this way. And at some point, there's a signal, and I don't know what that signal is, but whatever that signal is, the microtubules that have grabbed onto the kinetochore begin to pull back towards the centrosome, and that's called anaphase. It's very dynamic, and you can see this in real time. Late in anaphase, right, you have the chromosomes separated, and the last step is telophase, where in telophase you start to begin the process of regenerating a nuclear membrane, which basically means the ER comes back and surrounds the nucleus, and the cells are ready now for cytokinesis. So this is a great video. I love this video. It's specifically looking at a blood lily, which is a plant cell. The chromosomes in these plant cells are huge, and you can see very easily this process. So watch and see what you can see. Okay, right now you can see some chromosomes, and they're clearly not lined up anywhere, but they're going to start jiggling around, jiggling around, jiggling around, and they get pushed and pulled, pushed and pulled. You can't see the asters, you can't see the microtubules, but okay, they're getting close to being lined up on the metaphase plate. It's not perfect, but bam, right before that, there was a signal that said pull. All right, and this is a plant cell. Do you remember what I said to you about plant cells? Plant cells form a new membrane between the two new daughter cells, and that's basically membrane that's coming off of the Golgi is then placed between the two cells and creates the cell membrane between the two new plant cells. Let's watch that again. And so if you watch, you can see the chromosomes, you can see sister chromatids, right? They start to move around, and this is basically pushing and pulling by the microtubules, and eventually they end up close to the metaphase plate, and the metaphase plate really is this place right in the middle, and then bam, right there, there's a signal. The signal causes the separation of the sister chromatids. The microtubules undergo catastrophe, and the chromosomes are pulled as far apart as possible. And this ensures that you don't end up with too few chromosomes on one side or too many chromosomes on the other side. So I told you, I alluded to this, we would get back to this, and the beauty of this is you know a lot about microtubules now. You know that they're dynamic. You know that they can form spontaneously, and then they can undergo depolymerization through the process of catastrophe. This image shows you that the entire spindle complex is composed of 3 sets of microtubules. So on the top, you have a cartoon structure, and in the bottom you actually see a real EM picture. And why I wanted to show this to you was, I hope what you can get from this is that the microtubules that emanate from the centrosomes, from the asters on the opposite poles of this cell, go in both directions. There's a lot of microtubules facing out towards the edge of the cells, and they become important for the process of getting the two new nuclei that are going to form to be as far apart as they possibly can be, so that we don't have separation of two cells inappropriately. Okay, so there's 3 sets of these microtubules. I just blew this up for you so that you could see it better. The first one on the left are called astral microtubules. Those astral microtubules emanate basically from the centrosome towards the periphery of the cell. And we're going to talk about them in just a minute. They actually attach to the cell membrane with a motor protein. Okay. Now there's 2 sets of microtubules that emanate towards the center of the metaphase plate. One of them is called the Kinetochore Microtubules. Those are the ones that actually attach to the kinetochore on the chromosomes. And remember, the kinetochore is a set of proteins that bind to, do you remember? They bind to the centromere on the chromosome. Remember, the centromere has heterochromatin. It doesn't have a lot of genes in it, so it's not being transcribed. So these protein complexes load on top of the centromere, and the kinetochore then allows for attachment to the microtubules. All right, the other set of microtubules are really important. They're called overlap microtubules. They're shown in red on this diagram. And what they're showing you here in black, the little black balls, are actually motor proteins. So the overlap microtubules are connected to one another by motor proteins. And what this means is you have a small motor protein that binds to both this microtubule and to the microtubule facing it. And so they can only be separated by a very small distance. And what's really important about these is that they are plus-ended microtubule motors. Which ones are those? Kinesins. What are the minus-end microtubules? Dyneins. So at the middle of the spindle complex, you have motor proteins on the overlap microtubules that are kinesins. And notice you also have motor proteins that are on the spindle complex at the very close to the centrosomes. Those motor proteins are negative end motor proteins. They're dyneins. Okay, so where are the negative ends? At the centrosomes. All right, and those motor proteins on the overlap microtubules actually generate this particular structure for the spindle complex. How do they do that, you might ask? Well, it's all about how quickly these microtubules get polymerized. I do like this graph, which means it could show up somewhere. The two lines in this graph talk about the half-life of microtubules at different times of the cell's life. So during interphase, right, the 23 hours that cells are normally doing their normal process of G1, then they go through replication, then they go through G2. It's just the way life is in a cell, right? They're constantly replicating, and during those 23 hours, the half-life of a microtubule is approximately 5 minutes. So that's actually pretty stable, even though we're talking about them being polymerized and depolymerized. Because if you look at that and compare it to the other line, the T1 half, the half-life of a microtubule during metaphase is 15 seconds. That means they're being made, they're being rejected. They're being made, they're catastrophizing. So this is a constant process, and it's happening very, very quickly during metaphase. What regulates that? A lot of proteins that we don't talk about. Okay, there are microtubule-associated proteins, MAPs, okay, and there are another set of proteins called catastrophins. Eek, what kinds of names are these? These are all proteins that associate with microtubules to keep them either stabilized into their long structure or to help increase the rate of catastrophe. And these are all regulated by a bunch of proteins called cyclin-dependent kinases. You know what kinases do. These are serine-threonine kinases. These are going to phosphorylate proteins. The phosphorylated proteins either lead to stability in structure or to instability in structure. It just depends on which ones get phosphorylated and when. And these cyclin-dependent kinases are turned on during mitosis. All right, I alluded to you that these microtubules actually form the structure that is the spindle complex. And I think of this sort of as a game of pickup sticks. If you throw a bunch of pickup sticks down, and these are your microtubules, and you were to have a motor complex that has a double head, all right, so you know, all right, that kinesins have, right, they have two heads and they walk along, right, and they move processively. Dyneins are the same thing. Okay, so now what you have is you actually have two of these linked together. And that's what these motors are that are associated with the spindle complex. So in this case, you have the black motors shown here are dyneins, and what they want to do is they want to march to the minus end of the microtubules. Well, if they're attached to two microtubules at the same time, as the two microtubules, right, if the proteins are at the ends, at the plus ends, sorry, you can't really see that, if they're at the plus ends and they move to the minus ends, it causes the minus ends to come together and the plus ends can now be separate from one another. And what it shows on the right side is that's really how this generates the spindle complex. You have these microtubules that are out there floating around and they get concentrated at the centrosome. The plus-ended microtubules, shown in B, are all about these overlap microtubules that generate, right, the rest of the spindle complex. And so they're plus-ended motors, and if you have a plus-ended motor, this side is trying to go towards this plus end, and this side is trying to go to this plus. Well, they can only get to the plus end, right, if you move this one back and you move this one, because they're only this far apart, right? So if they're here, the only way to get this to happen is to push the two spindle complexes apart, and right, they can only end up like this. So they push the whole spindle complex further away, and what's good about this is it causes the entire metaphase plate to be elongated. The metaphase plate stays right in the middle, but when the chromosomes get pulled, they get pushed further and further apart so that when the contractile ring comes along, there's no stragglers, there's no ends of chromosomes that are going to get cut off in that contractile ring. Okay, so this picture is really showing you that you get these microtubules emanating from the two centrosomes. They overlap in the middle with the kinesins, and that those overlap microtubules help develop the whole spindle complex. And once again, as they get pushed further apart, because the motors, right, are trying to reach the end. If the motor's here, right, and it's trying to get to this end, the only way for it to get to this end is to pull this back, and this one to be pulled back as well, and that pushes the entire complex further apart towards the periphery of the cell. Okay, this picture is just showing you the kinetochore. It's a set of proteins that are bound to the heterochromatin that's at the centromere region of the chromosome. And the microtubules, there's some question as to whether they actually attach to the kinetochore proteins or if they actually attach in some way to the kinetochore proteins like this, so that the end of the microtubule is here and it can polymerize and depolymerize still, and right, it moves along on the kinetochore bound by some peripheral proteins that are binding to the outside. I like this EM picture because it really does show you the chromosome is the dark material, and you can see the plate, that's the kinetochore, and you can see the microtubules attached, right? In this case, the kinetochore microtubules are pulling the chromosome away from its sister chromatid. Alright, and this is showing you the actual microtubules, the DNA in blue, so the microtubules are in green, the DNA is in blue, and pink is actually the centromere, the kinetochore proteins are stained with pink. So what you see is little pink dots, and each of those has a kinetochore microtubule bound to it, and it's pulling these chromosomes apart. Okay. This picture, sorry, let me go back. This picture is clearly when they're located on the metaphase plate. This means that you have a chromosome, right, with sister chromatids, and it has one centromere. It has one kinetochore complex. And so you have microtubules from both sides are bound to that same centromere or kinetochore complex. So they bind on both sides and they do this little push me, pull me thing, and that jiggling ends up getting them on the metaphase plate. And then when the signal occurs, bam, you go right from panel A to panel B pretty quickly, right? So anaphase is the separating of those chromosomes. And you can see some of these chromosomes have very long tails, some of them have very short tails, and that just has to do with where the centromere is on each of the different sister chromatids. Okay, so what do you need to know? you need to know that during S phase, you duplicate the centrosomes. So you get 4 centrioles, right? And you get duplicated DNA. That's during interphase. Interphase is G1, S, and G2. Okay, during basically the next one that really matters is metaphase, and at metaphase you have the chromosomes lined up due to the pushing and pulling of the kinetochore microtubules on the kinetochore of the sister chromatids, and at anaphase you have the pulling of the kinetochore microtubules. What happens to those overlap microtubules? They actually stay there, and that's when they do this lengthening. They lengthen out and push the entire newly formed, what's going to end up being the newly formed nuclei, to further ends of the cell so that the contractile ring doesn't make a mistake. Alright, and this is another picture just showing that. You still have in anaphase, I don't know why we have anaphase A and anaphase B. Basically, you have anaphase is pulling of the chromatids and the pushing of the centrosomes further apart. And the last thing that happens is you actually have the astral microtubules are attached to the plasma membrane via a negative end motor. So they have a dynein attached to the plasma membrane and to the microtubule, and if it's trying to go to the minus end, where is the minus end on the astral microtubules? At the centrosome, right? At the centrioles. So it actually pulls the entire complex. Right? So if the microtubules are emanating out here, the motor causes it to go towards the minus end, but it's attached to the plasma membrane, so it pulls the entire centrosome and the newly pulled chromosomes further to the edge of the cell. All of this is to allow for the contractile ring to come in and not separate any chromosomes inappropriately. So you have 2 different pushes and pulls. You have pushing, right? These 2 are pushing apart, and the pulling of in this picture, the red are the plus-ended kinesins that are on the overlap fibrils, the overlap microtubules. The black motor proteins are the minus end motor proteins that pull the centrosomes towards the poles of the cell. Okay, the last part of this, cytokinesis, right, divide and thrive. So I don't know why we say this, but right, this whole division, where are the actin filaments that are doing cell division? Are they on the outside of the cell, or are they on the inside of the cell? Where do we generate actin? Actin is on the inside of the cell. It really does look like the cell is being pinched from the outside, but it's being pinched from the inside. It's like having a waistband, right, on your pants that is being tightened around you, and it's trying to pinch you in half. And that is due to the actin and myosin motor proteins. So I like this picture once again. So on the left in A, they're showing you a diagram of what this looks like, and in panel C they did fluorescent staining of cells undergoing cell division. In red they stained for actin, and you can see this big band of actin between the 2 cells. And what's all the blobs of actin around the edge of the cell? That's cortical actin. Okay, cortical actin doesn't just go away, but it does remain there, and it's going to help form the cells after cell division. What do you think green is staining for? So if red is the actin, green are the myosin motor proteins. So you can really see very, very nicely the co-localization of these motor proteins with the actin contractile ring. So that's actually happening on the inside of the cell. It's hard to tell in this EM picture what's happening. Outside the cell, there's no actin. Inside the cell, there's actin, and it's actually being pinched in, causing the 2 cells to separate. And this is an even better picture. So in A, that's a scanning electron micrograph. It shows the 3D structure of these 2 cells, and on the bottom you see the transmission electron micrograph. You see the black banded line, and that black band is where the actin and myosin motors really are. all of the microtubules are still out there. Those are the overlap microtubules. So the overlap microtubules actually get pinched off, right? And that's what closes the two cells from one another. And that's it. That was really fast. I do go much faster when I don't have an audience. I hope that helps you, and I will be doing the next lecture for you as well before the weekend is over. Have a great weekend. Enjoy your turkey, and there's going to be a quiz on the two of them. I'll make it one quiz for both. Okay? Have a great day.